voice audible?
Yes, ma'am. Yes, ma'am. It is audible.
Okay, okay. So yeah, can you see my PPT?
Yes, ma'am.
Okay, fine. So let us start the class. Please everyone mute your microphones.
Yes.
Yeah. Thank you. So today we will be discussing about cell membranes. So especially the plasma membrane. Okay, so that is the main membrane which surrounds the cell. So before going into the topic, first we will look into what are the lipids aggregate. So lipids are non-soluble in water, so that we all know. So these lipids, when added in water, they will form aggregates. Like for example, if you drop a oil on in some a glass of water, so what happens? It is insoluble, and all the oil molecules, initially though they are separated, they come together to form a single layer or single bubble kind of thing. So that means the lipids will form aggregate structures in water. So what kind of structures they take? Okay, when we look normally, we don't find any structures, but when we observe under microscope, they can form different structures. So based on the type of lipids present and the concentration of lipids, so they form different structures like micelles, liposomes, and bilayers. So which are now in scientific research, they have utmost importance because they are liposomes especially. So they are used in the drug delivery and also to carry something like nanoparticles or proteins or enzymes. So for many purposes, these liposomes are being used. So this is a micelle. So whenever a lipid, that too amphipathic molecules. So what do you call amphipathic means? Amphi means two. Pathic means like which are like if you have hydrophobic, hydrophilic molecules. Now here if you see the blue color ball which is present, that is polar head. Okay, that means polar end of the fat which is soluble in water. Whereas this yellow color tail, that is hydrophobic in nature. That means the fatty acid tails are non-soluble in water. Hence they are not polar. So non-polar. So here you have polar group and non-polar group in the same molecule. Hence we can call them as amphipathic molecules. So they have large head and small tail. So here they are like fatty acids, okay, which are linked. So each micelle has form a few dozen to few thousand lipid molecules. So whenever we add such lipid molecules into water or when they interact with water, they immediately get a shape like they will surround like this because these tails are non-polar, right? So they cannot interact with water. So immediately they try to escape the water. They don't want to get exposed to water. So immediately all the polar groups will form a surrounding structure like this, and all these non-polar molecules will be inside this shell. Okay, so this kind of structure is called as micelle. So in this we can have few dozens of lipid molecules or few thousands of lipid molecules. So this aggregation occurs when the concentration of molecules, lipid molecules, are higher than certain threshold molecule. So always there will be a threshold. So for anything, so if there is the concentration is more than a threshold, then it can form aggregation. Okay, so they are wedge shaped. If you see individually, they are like wedge. Wedge means the upper portion is wider and it comes to narrower when it comes to the bottom portion. Okay, then vessel or liposome. So here we can see you have polar heads surrounding the non-polar one, and inside also you have a layer. Okay, so they are small bilayers. Now when you see the top one, So that is not a bilayer. It is a single layer. If you understand from the structure. So here only one layer of heads, then below tails are there. But if you see in the liposome, it is bilayer. So you have a head group here, then tail group. Again, here one head group and one tail group. So these two tail groups are like back to back. Okay, they are like both of them, they form a central hydrophobic region where they don't expose to the water. But below are the like innermost portion is again polar head group. So here we can see some water interaction with the water. So outside it is exposed to water and inside also it is exposed to water. And in the center portion, it is like it is hydrophobic. It is not interacting with the water. So small bilayers which spontaneously seal into spherical vesicles, and vesicle membranes can contain artificially inserted proteins. So we can also prepare, like we can design what kind of lipids or proteins, how it should look like, and what kind of molecules we have to insert. And the central aqueous activity can enclose dissolved molecules. So you can see any dissolved molecules here. And they are artificial carriers of molecules. So like drugs, I told you, like liposomes are, there is a lot of research going on, like for acting them as a drug carriers, or like especially anti-cancer drugs or such that. And the vesicles fuse readily with cell membranes or with each other. So they can fuse with the other membranes or within themselves also. Okay. Now we come to membrane bilayer. So if we see the liposomes or micelles, they are now artificially prepared. Means they are in smaller scale, like a small oil droplet. Okay, so they are nothing but smaller scale, but naturally present. Now we are exploiting those structures to apply them in biomedical field. But the larger one is membrane bilayer, which is seen in all types of cells. So this plasma membrane is a lipid bilayer, and it is present in all types of cells. Okay, irrespective of whether it is bacteria or plant or animal or any cell. Okay, so what are the features of this membrane bilayer? We will look into it. So it contains two leaflets of lipid monolayers. So it is similar to the liposomes, where we can see, so two layers here, the top layer and this is the bottom layer. So in the top layer, you have a head and you have fatty acyl tails. And the same thing in the inverted direction, you can see the heads and tail groups. Okay, so the hydrophilic head groups interact with water. So whatever the top portion, if you see the cell, so the outer portion of the cell is usually dipped in, like it is exposed to water, right? So because in our body, like cells are surrounded by water. Water in the sense we have different fluids, like either plasma, serum, blood, or lymph, or many types of fluids are there. So the cell, outer portion of the cell will get exposed to the water, that is aqueous environment. And at the same time, inside of the cell, that is where the cytoplasm is present, again that is a aqueous environment, and that inside environment is also exposed to water. Hence these polar head groups are present outside and inside of the cell, and in between we have hydrophobic fatty acyl tails, and one leaflet faces the cytoplasm. So the bottom portion will like they get exposed to the cytoplasm, and another leaflet exposes to the extracellular space. Okay, so this is how the bilayer looks like. So the plasma membrane is a lipid bilayer, and the name leaflet means the outer portion, that is a head and two tails. This layer is one leaflet and the bottom one is another leaflet. Okay. So first of all, what are the membranes? So they are lipid based structures which form sheets, And not only lipids, here we can see proteins, carbohydrates, and also cholesterol. And some membrane lipids and proteins are glycosylated. Okay, there will be modification. Glycosylated means sugar molecule or carbohydrate molecules will get linked to the lipids and proteins. So when a sugar molecule is linked to a lipid, then it is called as or lipopolysaccharide, okay, or glycolipids. If it is a polysaccharide, we can call polysaccharide. If it is a simple sugar, like glycolipids. And if it is a protein, then glycoproteins. That means a carbohydrate molecule is linked to the protein. Now, all cells have cell membranes which separate the cell from its surroundings. So, what is the main purpose of the cell membrane? It is because there will be a like distinguish, like you can see if you take our house as an example. So, we will have different rooms. So, how you distinguish a room? It is a single block only when it is constructed, but it is made into rooms by constructing walls around it, so that you are giving a boundary to it. In the same way, a cell will have a boundary so that it will get separated as a cell. It is given as a name as a cell, so it will separate the external environment from internal environment. And eukaryotic cells have various internal membranes that divide the internal space into compartments. That means within the cell also we have various organelles. And each organelle, so in the last class, we have already discussed membranous organelle and non-membranous organelle, like mitochondria, or if you see chloroplast in plants, or endoplasmic reticulum, nucleus. So we have where we have membranes which are surrounding those organelle. So that means even within the internal space also it is discriminating or differentiating the structures from the others. So this is how it looks when you see under electron microscope. So this is endoplasmic reticulum, this is nucleus. So this is the nuclear membrane. Now we can easily detect this as a nucleus. If there is no membrane, we cannot detect. Everything looks similar. Okay. So this is mitochondria, which is surrounded by mitochondrial membrane. And these are the secretory granules which can secrete some proteins. So even they have their membranes. So like that, membrane will delimit the structures. So what are the functions of the membranes? So first is they will give the boundary to the cell, so we can distinguish, and allow import and export. That means importing means like if any molecule has to enter inside, then that will be checked by this plasma membrane. And also exporting, if any secretory molecule or anything has to go out of the cell, then that also will be checked. So we'll look into the examples like importing, selective import of nutrients. So I think you all know operon concept. So operon is nothing but a group of genes which will utilize the lactose. So you have operon, like that you have. That means when a bacteria grows in a particular nutrient medium, so that will acquire the nutrient. If there is a glucose or lactose or whatever it is, that has to come inside the cell. So that coming inside the cell is import. That means the nutrients has to import the cell. So here that importing is again controlled by the plasma membrane. At the same time, the waste or toxins, some secrete some toxins like enterotoxins, mycotoxins, and some bacteria, they secrete antibiotics to kill other bacteria. So such kind of products which are secreted by the bacteria again has to go out. And this is in a selective way, that not everything comes inside and not everything goes outside. So it is highly controlled. That's why we call plasma membrane as a semi-permeable. Permeable means anything can come. Okay, it is readily it can come, but it is semi-permeable. That means not everything can come inside and not everything can go readily. Okay? So, and the next point is they retain metabolites and ions within the cell. So if there is no boundary, what happens? So everything can, it is simply, there is no limit, right? There is no boundary. Anything can come inside and anything can go outside. So the metabolites or ions which are generated due to metabolism or anything, so that cannot be retained in the cell. So this plasma membrane, they retain the metabolites and ions within the cell. And the next is they sense external signals and transmit information into the cell. So if there is any signal or stimuli from outside the cell, like for example, in the case of immune system, if any antigen enters, so definitely what happens? That signal goes to the cell, like immune cells, like phagocytes, neutrophils, all this. So they will get activated, right? So in the same way, if there is any like death signals also will be there. If the cell has, if something goes wrong in the cell metabolism, or that cell has become cancerous and it has to die, then immediately it will get a death signal. So like that, there will be some signals, and if there is any signal for division of cell, so that signals are received and that information is transmitted into the cell and then provide compartmentalization within the cell. So that's what, like we have to make the cell or organelle or cells, we have to compartmentalize. Okay, is clear distinction is important. They separate energy producing reactions from energy consuming ones and keep proteolytic enzymes away from the important cellular proteins. So what happens? See, in our cell, we have proteolytic enzymes. At the same time, we also have proteins. Then there is definitely a chance that these proteolytic enzymes, so what are proteolytic enzymes? Which will cleave the proteins. So they can act on the self proteins. But that is not happening. This is because the membranes are separating these two components. And even energy producing reactions and energy consuming reactions are also separate. And they produce and transmit nerve signals. This especially in case of neurons. So we have myelin sheath, axon. So axon is surrounded by myelin sheath. Again, there comes the membranes. So whatever the signals or the what you call synapse, so that signals are transmitted and produced by the membranes, and they store energy as a proton gradient. So in the mitochondria especially, during oxidative phosphorylation or like that, the energy has to be stored by the proton gradient, so that the ATP is synthesized. So the ATP synthesis all takes place in the membranes of the mitochondria. So these are various functions, important functions of the membranes. Then common features of the membranes. So they're all sheath-like flexible structures. They're all like in any organism you see, they look like similar. They are thick. And the main structure has two leaflets of lipids. That's why we call it as bilayers. But in archaebacteria, so archaebacteria is a type of bacteria which is like ancestral form. That means initially originated from. So there we can see only monolayer of bifunctional lipids. That means only monolayer, but the lipids are bifunctional. They can perform two functions. And they can form spontaneously in aqueous solutions and are stabilized by non-covalent forces, especially hydrophobic effect. So there is no much reaction required to form bilayers or membranes. So spontaneously, when we add in the water or aqueous solution, they will form the layers. And there are no covalent forces because that has to be flexible, right? So if the cells are rigid, then they cannot perform what functions they are performing. Therefore, they require some flexibility. So for this, there should not be any covalent interactions. So here their hydrophobic effect will be generated. And the protein molecules span the lipid bilayers. So we have some protein molecules also in the lipids. And these two layers are asymmetric in nature. They are not the replica of one or each other, right? So if one leaflet is looking similar, like one type, and the other leaflet will be of different. That means in what way they are different, they look similar, but the lipids which are present or the molecules which are present inside will be different. So some lipids are called as inside and some lipids are preferably outside. That's what the composition will be different. And carbohydrate moieties are always outside the cell. So there are some carbohydrate entities which will be present. And they are electrically polarized. So that means the charge will be different in two layers. And the fluid structures are two-dimensional solution of oriented lipids. It's a two-dimensional structure, not the three-dimensional. So the most accepted model to explain the structure of the membrane is fluid mosaic model. So it was proposed by Singer and Nicholson in 1972. And lipids form a viscous two-dimensional solvent into which proteins are inserted and integrated more or less deeply. Okay. And integral proteins are firmly associated with the membrane, often spanning the bilayer. So we have different types of proteins. So we have peripheral proteins, transmembrane proteins, and protein associated lipid associated proteins. Now the peripheral proteins which are weakly associated and they will be present only on the outer surface of the membrane and they can be easily removed. There is no any covalent interaction or like that. They are non-covalently linked and some may be attached to the lipids also. But the integral proteins, they usually transmit from outside to inside. So we have extracellular domain, then transmembrane domain and cytoplasmic domain. So this is how it looks like. So if you see, this is the outside environment, this is the inside environment. So here you have glycolipids. That means to the lipids, if we have the carbohydrates or sugar molecules, then that is glycolipid. And if it is oligosaccharide, so if you see, these are glycolipids where the sugar molecules are linked, and in the oligosaccharides we have many sugar molecules. So you have oligosaccharides, polysaccharides and monosaccharides. You might have aware of the terms. So saccharide means a sugar, okay, and monosaccharide means a single sugar moiety, and if it is a oligosaccharide, many sugar molecules, and if it is polysaccharide, you have hundreds of sugar molecules. So these are the carbohydrates which are present, and these are the proteins, GPI anchored protein. So this, when the protein is anchored to the carbohydrate here, so they are nothing but the anchored proteins we can call. And this is the lipid bilayer. And you have peripheral proteins where proteins are just linked to the membrane, non-covalently attached. Okay. But here if you see, there are peripheral proteins which are covalently linked to the lipid, not directly. Okay. That means it is protruding inside the membrane. And these are the phospholipid heads, polar heads, and these are the sterols, orange color ones. Or here in eukaryotic cell we have cholesterol, and this is transmembrane protein or integral protein. So if this is outside, this is called the portion which is present on the outside of the cell is called extracellular domain, and the one which is spanning inside are transmembrane domain, and the one which is present towards the inside of the cell, that is cytoplasmic domain. Okay. Okay. So the composition of membranes. So the lipid composition of membranes is different in different organisms. That means not all organisms or all tissues or all organelles will have the same type of lipids. So in eukaryotes, we have phospholipids. That means the lipid molecules having the phosphate moiety. And the ratio of lipid to protein also varies. That means for how much amount of lipids, how much amount of protein is required, that also varies from different organism to different organism. And what type of phospholipids? So we have phosphatidylserine, phosphatidylcholine, phosphatidylethanolamine, phosphatidylinositol. Like that, we have different types of phospholipids based on the functional group which is present. And that also varies from one organelle or tissue to organism. Okay, it can vary. And the abundance and type of sterols also vary. So here you have cholesterol in eukaryotic cell. But in bacterial cell, we don't find cholesterol. So cholesterol is completely absent. So there we see hopanoids. They are also sterol-like molecules, but not exact sterols. Okay. So that's what prokaryotes, they lack sterols. Whereas cholesterol is predominant in the plasma membrane, but that is absent in mitochondria. And in plant chloroplast, we can say galactolipids, but they are not present in animals. So such kind of differences are seen. So here you can see the type of lipids present. In human myelin sheath, in neurons, 30% of protein, or 30% of phospholipid, and 19% of sterols. So the major sterol which is present is cholesterol, and other lipids are galactolipids and plasmalogens. But that is not the case with the other. If you say mouse liver, the components have changed. The percentage of protein has changed. Phospholipid has changed, and sterols also. Sterol type is same, but the percentage is different. So another, the best example is E. coli in the prokaryotes. It has 75% of protein, 25% of phospholipid, but sterols are completely absent. Right? So like that, you have like different types of lipids, proteins, and sterols, and their composition also varies. If you say in plant, you have cytosterol instead of cholesterol. Right? In yeast, we have ergosterol. In paramecium, we have stigmasterol, like that. So when we see the RK, okay, in RK, L-glycerol is present when D-glycerol is present in bacteria. So you have differences between the archaea and bacteria also. And there are some unique fatty acids which are present. They are branched isoprene chains in archaea, whereas unbranched fatty acid chains in bacteria. So the head group and the tail groups. So the tail groups are fatty acid chains, which are not branched. You can see only two chains. But in RK, we see isoprene chains instead of fatty acid chains, and they are branched. And the linkages. We have ester linkage in bacteria and ether linkage in RK. And the topology, that means the structure, is monolayer in RK, whereas bilayer in bacteria. So this is the example. Sulfolobus solfatariacus and their relatives, they are present in volcanoes, hot springs, so they can withstand at least 75 to 80 degrees centigrade, and the environment is acidic. The pH is 2 to 3 here. So therefore, it based on the environment where they are surviving, so the composition varies. So here we have isoprenoid tetraethers with unique alcohols here. So the structure will vary. Yeah. So as I told you, this membrane bilayers are asymmetric in nature. That means both are not similar. So they have different lipid compositions, and the outer leaflet is more positively charged, whereas the inner leaflet is negatively charged. So this phosphatidylserine is usually present inside the leaflet, inner leaflet you can call. But only in some condition, this will come outside. So that is again a signal. So that means when a cell has to undergo apoptosis, that is a programmed cell death, the phosphatidylserine, which is present in the inner leaflet of the cell, will come to outer leaflet of the cell. And this marks the destruction of the cell. So that is a signal. And there, if in platelets this happens, then the clotting of blood will be activated. So that means, so how important the symmetry are. So if one lipid comes here and there, then completely the signaling will change. If somebody has any doubts? Okay. So you can see here, so phosphatidylethanolamine is more in the inner leaflet. And phosphatidylserine is also inner. Phosphatidylinositol, phosphatidylinositol 4-phosphate, phosphatidylinositol 4,5-bisphosphate, and phosphatidic acid. So these are all more in inner leaflet. But phosphatidylcholine and sphingomyelin, these two are more in outer leaflet. Okay. So what is the membrane composition? How it looks like means, so here you can see sphingolipids and cholesterol. They are present here. And phosphatidylethanolamine and phosphatidylserine are present in the inner leaflet. So these are different combinations. So phosphatidylcholine, sphingolipids and cholesterol, if they are present outside, so always the phosphatidylethanolamine and serine, they will be inside. And if this comes outside, if you can see here, if it comes outside, then that will trigger the signals. Okay. So now we have discussed, like there are different types of proteins, like integral proteins, transmembrane proteins, and peripheral proteins, or lipid anchored proteins. Now what are the functions of the proteins? They act as receptors in the detection of signaling from the outside. So that means these, especially these transmembrane proteins, which will be passing from outside the cell to inside the cell. So therefore that can act as a receptor, so that the signal will be transmitted towards the interior of the cell. So what kind of signals it will carry? First is light. If that means in case of eye, so we need a signal, so that our eye receives a signal from outside that if there is any light or darkness, so that has to be received by the internal end of the eye. So here opsin is the protein. Then hormones like insulin receptor, so they will bind specifically to insulin. And then acetylcholine receptor, which will receive the neurotransmitters like acetylcholine. And then taste and smell receptors like pheromones. These are the signals, so which will be received by the receptors. Then channels, gates, and pumps. So apart from the signaling, these proteins also act as the channels, gates, and pumps for the permeability of small molecules. So for nutrient movement, like maltoporins. Maltoporins are the proteins which will form a gate for or a channel for transport of nutrients. Then potassium channels for transport of potassium ions. Then serotonin reuptake protein, which will transmit serotonin, which is again neurotransmitter. Then enzymes. These proteins can also act as enzymes, especially in ATP synthesis. In mitochondrial membrane, we have F0, F1 particles. So I told you in the last class, F0, F1 particles, there is an enzyme F0, F1 ATPase. So this 8 or the other name is ATP synthase, where the ATP synthesis takes place. And some acyl transferases, which will help in lipid biosynthesis. Okay, like this, various proteins in the membranes, they function. So the types of proteins, as I already told you, like these are the peripheral proteins, which are just linked. So if there is any change in pH or chelating agent or urea or carbonate, this protein will be released from the attachment. That means you can simply remove by simple change of acid. There is no harsh treatment or something like that. Simply we can remove. And then amphitrophic proteins. So these, through biological regulation, also we can remove or we can attach. Then here you have a GPI anchored protein. So that means here you have carbohydrate to which you have protein which is linked. Here you have phospholipase C. So here the phospholipase C will cleave this bond, and then this protein along with the sugar molecule or glycan will be removed. So here you need enzymes for treatment. For integral protein, so it is very strong. So you need a treatment with a detergent. So that strong detergent, so that the entire portion along with some lipids will be removed. So this is how we can remove the membrane proteins from the membranes. So peripheral membrane proteins. So peripheral membrane proteins, they associate with polar head groups of the membrane. So to the polar head groups, these proteins will be linked. They are loosely associated with the membrane through ionic interactions, and they can be easily removed by disrupting ionic interactions either by high salt or change in pH. And these purified peripheral membrane proteins are no longer associated with any lipids. So it is very simple thing we can remove the proteins. Then integral membrane proteins, they span the entire membrane. They have asymmetry like the membrane. So even like as the membrane, two leaflets are asymmetrical, even the different domains like extracellular domain, transmembrane domain, and intracellular domain or cytoplasmic domain, these three will be asymmetric in nature. They're tightly associated with the membrane. So if you remove the membrane, if you remove the protein, the membrane also will get removed. So the hydrophobic stretches in the protein interact with the hydrophobic region of the membrane. And they have to be removed by detergents, which also disrupt the membrane. And the purified integral membrane proteins still have phospholipids associated with them. So this is how they look like. So they will be inside the membrane. So we have 6 types of integral membrane proteins. So if this is outside and this is inside, we have type 1. So where the amino group is towards outside and carboxy terminus, C terminus is inside. Whereas it is reverse in the type 2, where C terminus is towards outside and N terminus is towards inside. And we have type 3 where, so it is multiple spanning. So one terminal will be on the inside and one terminal with the outside, and you have the spanning of this protein. And type 4, so you have many proteins which are like functioning together. So everything has a C terminus and N terminus, and you can see different, they together collectively they are functioning. And then type 4, there is no extracellular domain, right? Only intracellular domain is there, or transmembrane domain is small portion is there, and you have cytoplasmic domain. Then type 6, although the spanning is there, it is like somewhat structurally it is different from that of normal transmembrane protein. So you have different types of proteins, and the amino acids which present in the membrane protein cluster are different. Okay, so here the transmembrane segments have hydrophobic amino acids, especially like tryptophan and tyrosine. Okay, and charged amino acids are found only in the aqueous domain. So these are some of the examples of maltodextrin or OMPLA, FEP-A. So these are the integral proteins. They contain beta sheets rather than alpha helices. Now lipid anchors. So some membrane proteins are lipoproteins. That means the proteins will interact with the lipids. They contain covalently linked lipid molecules. Whether they can bind to long chain fatty acids, isoprenoids, sterols, or GPI, that is glycosylated phosphatidyl inositol. And the lipid part becomes a part of the membrane, and the protein is anchored to this membrane. So that can be a reversible process. We can also remove. This allows the targeting of the protein. If it is fixed to particular membrane through lipid or something, so it is easy to target. Like if you have GPI anchored, they are present on the outer face of the plasma membrane. So like this, how the lipid linked membrane proteins are present. So these are the lipid molecules where you have proteins which will be linked here. So here you can see this is a pink color. One proteins which are linked to the lipids, yellow colored lipids, right? So we have another term called farnesylation of proteins. So proteins can be targeted to the inner leaflet of the plasma membrane by farnesylation. That means we have a sequence which is CAAX. That means cysteine, then AA means any aliphatic amino acids, and X is methionine, serine, glutamine, and alanine. So here the primary sequence of the protein consists of the signature CAAX. If that particular signature molecule is there, that 4 amino acid small region, then that is called as farnesylation. That means the protein can undergo farnesylation. Farnesylation is catalyzed by the farnesyl transferase. That means the proteins, if you like the targeting of a protein to inner leaflet, that is farnesylation. That means when the proteins are synthesized and packed like by Golgi complex and all, then they will decide like where this protein has to go. Then if there is a CAAX group in this protein, then that will decide that okay, this has to go to the inner leaflet of the plasma membrane, right? So here you have an enzyme which will take this protein and keeps in inner leaflet of the protein. So that is farnesyl transferase. And those proteins which are not having this particular segment CAAX, they are non-farnesylated proteins, which they cannot be inserted in the membrane and therefore they are inactive. So these are especially important in cancer therapy. Okay, so like for RAS protein, so you have, that is another topic, cancer biology or this. So here the cell membranes are asymmetric. So that's what we have discussed, that every component of the membrane is asymmetric. So even the lipids are different, the proteins which are associated on one side are different from the other side, and lipid modification also can be different. So based on the lipid modification, the insertion of proteins will take place. Okay, and carbohydrates, they are present only on the outside of the cell. So what are the physical properties of the membrane? They are dynamic and flexible and can exist in various phases and undergo phase transition also. So that means in aqueous phase or like that, and even they can undergo change also. They are not permeable to large polar solutes and ions. Only small polar solutes and non-polar compounds can be passed through. Why only non-polar components? Because the inner region will be hydrophobic. Though the head groups are polar, the inner region is non-polar. Hence, the polar molecules cannot enter. Only small polar solutes, that too, we will have various ion channels, or we have gates or transporters through which they will be transported. And the permeability can be increased artificially if we can treat the plasma membrane with some chemicals. So membrane can assume different phases like gel phase or fluid phase. Depending on the composition and the temperature, the lipid layer can be gel or fluid phase. Gel phase means the individual molecules cannot move, whereas fluid phase means the individual molecules can move. So when there is heating, what happens? This gel phase will become fluid phase. And under normal temperature, the membranes will be more fluid in nature. And even the presence and absence of cholesterol also will affect the fluidity of the membrane. And also the bonds, number of double bonds in the fatty acid chains. So this is how it looks like. When they are ordered, okay, what happens? They will tightly get packed. And when the distortion takes place, there will be space. Therefore, the movement will take place. So membrane fluidity is determined mainly by fatty acid composition. If the fluid membranes, if the fatty acid chains are short and more unsaturated. Unsaturated fatty acids means more double bonds. So what happens here, whenever a single double bond comes, so just assume you have a fatty acid chain which is straight. At some portion, like maybe fifth lipid, you have a double bond. So wherever the double bond is there, there will be a small bend which comes there. And it will go straight. And again after some 10th position, again one double bond. Again it will bend in other direction. So that means instead of completely straight, now it will form a wider structure with more bends. As the number of double bonds increases, that will become wider. So therefore, the space between different fatty acids will increase. When it increases, what happens? There will be more fluidity. And at higher temperature, cells need more saturated fatty acids. Saturated means single bonds, which are having single bonds. When only single bonds are present, they are tightly packed, right? So just assume, like if you have different files, okay, and if you want to arrange them in racks and all files are straight, that they are not bulky, okay, what happens? You can pack them very tightly, right? Many files can be put in the rack. But at the same time, if one file is thin and one file is bulky like that, what happens? The space will, in the same rack, you can now keep only few files. And the space will, like the gap will increase between the different things. So the same thing here, if you have more number of double bonds, the space will increase for the each fatty acid, and there will be more fluidity there. At lower temperature, cells need more unsaturated fatty acids. So that means at lower temperature, we need more fluidity, whereas at higher temperature, to like to tightly pack, to maintain the integrity, we need saturated fatty acids. So these are sterols and hopanols. So these are the structures. So cell membranes of many eukaryotes contain sterols, like cholesterol in animals, phytosterols in plants, ergosterols in fungi. And but in prokaryotes they are absent. So instead they have hopanols or hopanoids also we can call. So this is the difference in the structure. Then membrane dynamics. So here, what about the movement? So the lipids can undergo lateral diffusion within the leaflet. That means within leaflet, they can move from one place to another. Within within a second, like in 1 micrometer, that can move. Okay. So like this, initially it was here. Now it has moved here. And also it can also move from one leaflet to other. So I told you, like phosphatidylserine is usually present in the inner leaflet, but whenever there is some apoptosis or cell death, it will go to outer leaflet. So this movement is mainly mediated by flippase, so that they are the enzymes. So this is called as flip-flop diffusion. So from one leaflet it is coming to another leaflet, so in the transverse direction. So here this is an enzyme mediated, this is flippases. Okay, there are some enzymes which are called flippases which catalyze this transverse diffusion. So flippases use ATP to move the lipids against the concentration gradient. Okay, so here you can see ATP is getting converted to ADP and PI. And when the energy is utilized, this will come to the inner leaflet. And here another flopase is there. So this will make them to other direction. So flip flop. You have scramblase, another enzyme, which can from one position to like this, diagonally also it can move. So how this like all these were studied? So these membrane dynamics are studied by a technique called FRAP, that is fluorescence recovery after photo bleaching. So this will help us to monitor the lateral lipid diffusion by monitoring the rate of fluorescence return. So that means if a lipid is tagged to a fluorescent protein, when it moves to another, here the fluorescence will be lost and some other place again it will be recovered. That means now based on the signal, it is like a signal torch. Okay, so in the same way, based on the fluorescence, we can determine how it is moving. So by that, the scientists have found that there is a lateral diffusion of lipids. So here you can see this is the cell and you have the membrane. So here we have added a fluorescent probe to label the lipids. So like this, you have fluorescent probes, red ones, small ones. And now, okay. Now, if you view the surface of the cell by fluorescence microscope, it looks like this. When the laser beam is added, so it will bleaches the small region. Bleaching means the fluorescence will be removed. Again, after some time, after unbleaching, that means if the laser beam is removed, these phospholipids again diffuse. The other phospholipids which are present in the other region, they will diffuse to the bleached area. So again, if you see, you can see the fluorescence. So here you see the center portion is completely white. There is no fluorescence. But after removal of bleach, you can see again small red spots you can find. That means the fluorescence is returned back. Okay. This is FRAB. So the same thing, how it in the larger region. Step 2, and here again it is recovered. Then we have membrane rafts. Membrane rafts are nothing but it's a cluster of glycosphingolipids. So the lipid distribution in the single leaflet is not random or uniform. You can see, and some portions you have thick lipid layers will be seen. See here you can see, not it is in a single, like in the same level. You can see a thick portion from here to here, green color, right? So they are called lipid rafts. They contain cluster of glycosphingolipids with longer than usual tails. Therefore, they are more order, of course, but they have specific doubly or triply acylated proteins. And they allow segregation of proteins in the membrane. So if kaolin protein is present in the membrane, so that will result in the curvature of the membrane. So like that, based on the composition, we can see. So other modes of membrane curvature are a protein with intrinsic curvature and with high density of positive charges. We can see the curvature. On its concave surface, interacts with negatively charged phospholipid head groups, favoring curvature of the bilayer. Or a protein with one or several amphipathic helices, which are inserted in one leaflet of the bilayer. Also, it can result in curvature. And protein with bar domains can polymerize into superstructures and favors to maintain the curvature. So these are different conditions where. And these membranes can fuse with one another. Without losing continuity, they can fuse. The fusion is spontaneous, or there can be any proteins or enzymes which can mediate the fusion. So the protein mediated fusion examples are, if there is any influenza virus which enters into the host cell, then that will fuse with the membrane. So how the phagocytosis all takes place, I will explain you. And or the release of neurotransmitters at nerve synapses. So neurotransmitters, so synapse is nothing but the junction of neuron between two neurons or one neuron and one muscle. So here these neurotransmitters are stored in the form of vesicles. So vesicles again will have membrane. So when they are released, these vesicles go and fuse with the synaptic membrane of the next neuron or the receptor. The receptors which are present on the membrane of the next neuron, and the two membranes will fuse. Therefore, they release the neurotransmitter. So this is the fusion. So these are the various examples of membrane fusion. One is budding of vesicles from Golgi complex. So how the vesicles comes out from the Golgi complex, and this vesicle has to fuse with the membrane. This is called exocytosis. If anything comes from outside to inside, this is endocytosis. So this membrane is fused with the vesicle. or viral infection where a virus enters the membrane and here the fusion takes place. And fusion of egg and sperm. So that is another kind. Even there the membranes, zone of pellucida of egg and the acrosomes which are present on the head of the sperm. So they will fuse. And the fusion of small vacuoles in the plants and separation of two plasma membranes during cell division. So these are different kinds of fusions, membrane fusions. So this is how the neurotransmitter release takes place. So this is the secretory vesicle where it comes and you have snare protein. So it is a protein mediated. So the neurotransmitter which is filled with um, neurotransmitter filled vesicle will come and fuse with the plasma membrane by proteins with the assistance of proteins. So here you have V snare, T snare. They bind to each other, zipping up the from the amino terminus and drawing the two membranes together. Now they both come together so that this membrane fuses with this membrane. After fusion, they come like they will form a pore, and these neurotransmitters are released outside the cell. So same thing here in a larger way. So here you can see clearly like this is plasma membrane, this is neurotransmitter filled vesicle. You have these blue dots are neurotransmitters. You have V snare and T snare proteins. Now these two will form a zip-like structure with the SNAP 25, and they will try to like get this new vesicle, secretory vesicle, nearer to the plasma membrane. Okay. Now these two will fuse. This is called hemifusion, where the inner leaflets of both membranes come in contact and they will fuse. Once the fusion takes place, it will form a pore here at the center. And from the pore, the vesicle contents are released outside the cell. Now we have another thing is very important is transport across membranes. So how the transportation across membranes takes place. So the cell membranes are permeable to only small non-polar molecules which can passively diffuse. That means they don't require any energy or any like assisted transportation. But the passive diffusion of polar molecules involves desolvation and high activation barrier. And the transport across the membrane can be facilitated by proteins. Like if we have, this is assisted transportation. We have transporters, permeases, which can help in transportation across the membrane. So here we can see, first is primary active transport. First is simple diffusion, where the non-polar compounds only across a concentration gradient, they will move. There is no energy or anything required. Simply they can move inside and outside. Then facilitated diffusion means you have electrochemical gradient. So here you have some gates, channels. So through the facilitated means it is facilitated by some assistance. Okay. Then primary active transport. So against the electron gradient, it has to go electrochemical gradient. Hence ATP is required. So ATP is cleaved to ADP and PI, and that energy is utilized here. Then ion. This is secondary active transport, where the ions will be transported inside and outside. Okay. And this is ion channel, like potassium ion, sodium ion channel, where the ions will be moving. And then ionophore mediated ion transport, where down the electrochemical gradient that will move. Okay. Now the polar solutes need alternative paths to cross the cell membranes. So that means the polar solutes, they can normally, non-polar, small non-polar molecules can move. But the polar molecules, because the inner region of the membrane is hydrophobic in nature. So these polar molecules requires an alternative paths. So where you have transporters, which will be like if you have glucose, so we have glucose transporter proteins. So GLUT protein, okay, like that we have. So there are three types of transport systems: uniport, symport, and antiport. Uniport means only one molecule can move in one direction. And symport means two molecules can move in the single direction, okay, simultaneously. Antiport means two molecules, they move in opposite direction simultaneously. That means if one molecule is coming inside, that is helping the other molecule to get outside. Okay, and this symport and antiport they can call as co-transport, because two molecules are transporting at the same time. So I told you glucose transporter, so GLUT protein, so that also helps. It is a transporter molecule which helps. No need to go in detail. Okay, but this is how the mechanism we can see. So this is a glucose molecule, D-glucose. This will come and bind to the transporter, where this will get interacted. And here the outside region, first the outside region is open. This glucose enters. So then it will close. The outside region will be closed. This means the conformational change takes place. Okay. And the inside, it opens inside the cell membrane, and this glucose is released inside. And again, the inside portion will be closed and outside portion will be open for the next glucose molecule. So this is how the transport takes place. So we have like multiple glucose transporters are also present, like sodium glucose transporter and a glucose uniporter. So both are they operate in epithelial cells in the opposite sides. Okay, so sodium glucose importer means when the glucose enters outside to inside, the sodium will move from inside to outside like that. So it is a antiporter, or it can be symporters also. Okay. So here, sorry, it is not an antiporter, it is symporter. You can see here, glucose molecule and two sodium molecules, both are entering the cell, epithelial cell. So symporter. So both are moving in same direction simultaneously. Then bicarbonate transporter is an antiporter. Here, so carbon dioxide, which is produced by catabolism, enters the erythrocyte, blood cells. And this carbon dioxide, along with water, in the presence of carbonic anhydrase, it will form bicarbonate, H plus ion, and Cl will be diffused. Now this bicarbonate will be released outside. So you have a chloride bicarbonate exchange protein here. So this chlorine enters inside. So here an antiporter system is functioning. Like chlorine is entering inside, bicarbonate is entering outside the cell. And here when carbon dioxide enters, bicarbonate is released. That means it is antiporter in nature. Two types of active transport. There are primary active transport and secondary active transport. So primary active transport means you have ATP, which is dissociated to ADP and PI, and this helps in the movement of this particular molecule towards outside. Whereas if in the secondary active transport, again with the same dissociation of ATP to ADP and PI, so this will be released outside, and same thing, another molecules also enters inside. So that means here first moving outside is facilitated by ATP synthesis, whereas again the same molecule along with other molecule can enter inside by symporter mechanism. So we have ABC transporters for ATP hydrolysis to transport the substrates. Then another is like the generation of ATP. So here you have proton transport and chemical energy of ATP. So energy of ATP hydrolysis can be used to drive protons through the membrane. So in the F0, F1 particle especially, we have an enzyme called F-type ATPase. So the energy of the proton gradient can be used to synthesize the ATP. So this happens in chloroplast and mitochondrial membranes. So this is the F0 and F1 particles, how they look like. So the below portion is F0 and the upper portion is F1. So these are the ATPases enzymes. So this F1 is present in the cytosolic portion and F0 portion is present in the mitochondrial matrix. So this acts as a motor F0, which will help in the movement of the F1 particle. So F1 has 6 subunits. So to alpha, beta, gamma and delta subunits. So which will be rotating. And during this rotation, the conformation of these subunits will change and the ATP will be synthesized from ADP and PI. This is mediated by the energy supplied by this proton gradient. Okay. So this is in cytoplasm where you have V0 and V1 subunits. Okay. So this is aquaporin. Aquaporin are again a channels where aqua means water. Porin is a channel. So through which the water molecules can be passaged. Then ion channels. See, ion channels also they require gradients for active transport. So here we have if the gradient, if more potassium, there will be 2 potassium ions inside and 3 sodium ions. That means when there are 2 potassium ions goes outside, 3 sodium ions comes inside. So that is the method how they transport. Okay. And based on the gradient, if there are less potassium ions outside or like that, the channels will open and close according to the signals. So we have sodium voltage gated channel in the skeletal muscle and sodium voltage gated channel in the neurons and in cardiac muscle also. Then calcium channels in neuronal, that is in neurons and retina, that is eye. Then polycystin 1, then potassium in neurons, which is voltage gated channels, and this cyclic GMP gated channels, which is present in the retina. It is not specific to any particular cation. Then we have acetylcholine receptor in the skeletal muscle, and we have chlorine channel in the lungs. Okay, CFTR, which is, so if there is any that if the channels get affected, then that can result in these type of diseases: cystic fibrosis, retinitis pigmentosa. Okay, like that we have different diseases which are caused by this defects in these genes which are coding for ion channels. So this is cystic fibrosis. We will look into one example. If there is any mutation in the ion channel in epithelial cell, it can result in the disease called cystic fibrosis. So here, this is the inside portion of the cell and this is outside portion of the cell. So the channel is closed, R domain is phosphorylated. So when R domain is phosphorylated, then there is no ATP which will come and bind. Okay, so this is R domain. Here you can see the bottom. And when ATP is hydrolyzed, what happens? The channel gets opened. And these ATP molecules will come and bind and the chlorine molecules will be passed outside. And after some time, the channels again gets closed. And now the R domain is unphosphorylated and no ATPs are bound. So that means how the closing and opening. So that is what. So if that defect is there, what happens is either if it is continuously open, all the chlorine ions will move out. Or if it is completely closed, there will be accumulation of chlorine ions. So both are difficult, right? So now, so today we have discussed about Like what are the functions of the plasma membrane? So what are the structures and what are the functions, in the sense each one in detail we have discussed. What kind of transport mechanisms and where they are involved. Okay, so if you have any doubts you can ask, and I will, meanwhile I will also share another PPT. Wait. Okay, so some points I will skip because they're already explained. So this is what I was discussing. So this is how I was discussing. Like when there is a double bond, you will see a bend here. So when there is bend, other fatty acid chain cannot come and fit here, right? So therefore the fluidity of the membrane will increase. But here if you have saturated fatty acids, you have, they are tightly packed and there will be no fluidity. Okay, and if the cholesterol molecules are also present, then they will also provide some space and fluidity will be increased. Okay, so most of them are explained already. So yeah, this is how the membrane fusion takes place. So I told you, like endocytosis, exocytosis, pinocytosis, all this. So whenever a virus or any bacteria comes inside, that will come and bind to the receptor. For example, here you have HIV, which comes and binds to the CD4, and CCR5 is the co-receptor. So this membrane is fused with the cell membrane. Okay, and that will be receptor. So that will be released inside the membrane. Okay, so even here you see, like from endoplasmic reticulum lumen, the glycoproteins will be released, and this will be packed by the Golgi complex, and the vesicle is released here, and that vesicle has to fuse with the plasma membrane, and only the protein which is present in the secretory vesicle, that will be secreted outside the cell, and that will again go and bind to the receptors which are present maybe in the other cells. Okay, then so this is the transportation, how it, yeah. So this is extracellular fluid where you have high sodium concentration and low potassium concentration. So what happens? This we have sodium potassium ATPase or transport, where in the cytoplasm we have low sodium and high potassium concentration. So this sodium comes here and this will be released outside. And after some point that will reach the saturation. There is no more sodium ions required outside. Again, there should be a movement of potassium ions from inside to low. So what happens again? The potassium ions comes inside and that will be released inside the cell. So how the sodium and potassium ions will be, sodium will be exported to outside and potassium ions will be imported to inside of the cell by ATP, energy provided by the ATP. Okay, so yeah, so phagocytosis, endocytosis, and pinocytosis. So phago means eating. So phagocytosis, cell eating. So that means whenever there is any particular bacteria or any some pathogen comes, so that will the cell will engulf. That means see how it is surrounded by the membrane, and that will be engulfed by a vacuole. This is called food vacuole, where it is engulfed. And this, when fuses with the lysosome releases some enzymes digestive enzymes. Then the phagosomes, so here comes the phagosomes also will be participating in immune reaction. So they will fuse and the digestion enzymes will digest the particular pathogen. This is phagocytosis. If you see pinocytosis means cell drinking. So here there is no solid material but the fluid or the solutes which are present. So that will be fused here and a vesicle will be formed. And this is receptor mediated endocytosis. That means, so this receptor will be, so this red color ones are receptors. So this receptors will guide the fusion of these particular coat proteins inside, and it will form a coated vesicle where the receptors are present inside the cell. Okay, so these are different types of membrane fusions you can see. So this is how, like, a bacterium is engulfed in the food vacuole, and also pseudopodium of amoeba, it looks like that. And this is pinocytosis. See, these are the real images where you can see how the vesicles are formed. So this is receptor mediated endocytosis. Okay, so this is all about plasma membranes and other cell membranes. So if you have any doubts, you can ask. Yes, any doubts?
No doubts, ma'am.
Is it clear, everything?
Yes, ma'am.
Somebody asked a
Ma'am,
question. Yeah, yeah.
will you please share the PPT?
Yeah, sure. Can you add me in the group? You
Okay,
asked
ma'am.
the admin, you have sent someone's name. I will just
I
share.
can share the link, ma'am.
Yeah,
I'll share the link. Will you please provide your phone number?
yes. So I have added in the chat box. So
Okay, ma'am.
you can just share me the link. I will
Okay, ma'am.
share there. Okay.
Okay, ma'am. I'll share just.
Yeah. Okay. Thank you so much.
Hello, Fatima, ma'am. Hello.